the members of our group work on zebrafish, on chicken, human cells, and mouse embryos at the same time. And so what we're really trying to address is why do mammals have such a hard time of regenerating their hair cells? Because the regenerating species such as chicken and zebrafish can do it very easily. And so there's evidence from Litau's group here, who's a member of our group, that in mice, some of the genes cannot be turned on as these mice get older. And so we're trying to, and so these are genes that should be turned on in order to make new hair cells. And so what we're trying to figure out is now what are the mechanisms that allow these regenerating species, such as the chicken and the zebrafish, to turn on these genes, and why can they not be turned on in a mouse? And once we describe these processes, we might be able to actually trigger these gene activations also in a mouse and trigger hair cells that way. And so what we're doing right now is in our respective model organisms or cell types or animals, we collect data sets. We're looking at how these genes are being turned on and turned off. And hopefully that will lead to candidates that we can then use to trigger or to develop therapeutics in mammals to trigger this regeneration in the mouse and then eventually in humans. And in zebrafish, the advantage also is that zebrafish, which might sound very strange to you, actually have these hair cells also in the skin of their body. So they're not just in the inner ear, but they also have these sensory hair cells in the skin. And so we can really easily image them and manipulate them and study how they regenerate. And so that's why zebrafish is a very good model. And mouse is an excellent model for studying development and regeneration, is because there are really lots of tools. Tools meaning ways to do experiments in these animals, because so many people are working with mice, and you can breed them and introduce mutations in genes and then see how hair cells do not develop properly. And so thereby learn more about these genes. So that's how we're working on these model organisms.
Our group is using gene therapy approaches to deliver genes that we know are important to make hair cells, and we are delivering these to animals, mice or guinea pigs, in which we have deafened the animals, and we are looking to see whether these methods of gene delivery are capable of bringing back new hair cells from the damaged tissue. Initial results are promising, but we have a very long way to go, but we are exploring different techniques and technologies of administering these tools to the damaged ear.
I'm a data scientist, and I work on big genomic data sets, which is a lot of what this consortium is generating. Individual studies tell us a lot about a specific system, but a lot of the things that we really want to know about can only be understood by putting together all of the data that the field is generating. So my group is, and the integrative analysis working group, are finding the algorithms and strategies to put together all of the data in one place to get bigger, more holistic answers. So for the last year and a half, the question that we've been tackling is, what really is a hair cell, and how do all of the different kinds of hair cells in the different species, in different conditions, in different hearing and vestibular organs differ from one another? And so we put together data sets that, from a genomic perspective, describe 28 different kinds of hair cells. And we built a very large database for what's unique about each of those, as well as what's shared. And we're working on a paper that really, I think, for the first time, gives us this detailed understanding of what sensory hair cells actually are. And over the next couple of years, we're going to take the same kind of strategy to understand the supporting cells that would be the target for the therapies to regenerate hair cells. So understanding what those look like in a healthy animal and a healthy human. And then finally, what the dynamic changes are that happen successfully during regeneration and what the blocks might be in conditions where it doesn't happen to complement the work that the other groups are doing.
So the GEAR, I believe a lot of the people here are aware of, is a tool for sharing of multi-omic data and giving users maximum ability to interact with the data, even if they're not bioinformaticians. The challenge that we had in the HRP has been that there are very high quality advanced data at great depth on which genes are expressed in which cells, whether it's the chicken or the mouse or the human or the zebrafish, either when the tissue is a normal tissue or immediately after exposure to a toxin that will kill the cells. And this is really interesting and important because we have the problem, the basic problem we're all studying is why are the hair cells in the human cochlea not regenerating, whereas in other species they are. Now, being able to look at all of these advanced data in one place it used to be impossible. And what the consortium has done over the years, it actually provided the lion's share of the support for this tool that allows individuals access to data and to be able to see all of these data side by side and to actually ask questions like, I think this gene might have an important role and look and see how it behaves across different tissues or a group of genes, or just look at a specific model, a group of genes that change according to one of the researchers in the consortium in response to injury. And the neat thing is that two things happened. The first thing is obviously it allowed us as a consortium to talk to each other, to see each other's data, to come up with new ideas and to formulate experiments. But even more importantly, it generated a tool that allowed the entire auditory field to actually share data and talk with each other. And so the GEAR now has more than 1,000 individuals, researchers in the field with accounts. The accounts are free to make. It has over 1,000 data sets, of which about 200 of them are organized in an accessible format according to topics. So it will be development, regeneration, by organisms. Some of the data sets are just by manuscript. And it essentially turned into this one-stop shop where every researcher in the field just goes ahead and when they have an idea, a question, they're reading an interesting paper, they go to it. So we have thousands of monthly entries to the GEAR by people. We don't track what they're looking at. And I think that this contribution of the HHF has transformed the culture of data sharing in our field and certainly advanced discovery. So we're very grateful.
Like when I first became involved with as the scientific director, I had been involved a bit on the scientific advisory board, so I was really familiar with the mission. And I actually come from a basic neurobiology department where there's nobody around me who, I have one colleague who's working on the ear, but there's a lot of people working on some other, issues like spinal cord regeneration. And so actually, one of the first things I did was I brought on to the board, the scientific advisory board, Jigang He, who works on spinal cord regeneration, and Seth Blackshaw, who's working on retinal regeneration. And so we talk to them regularly about how we can learn from their efforts. It's been incredibly helpful because, for instance, Jigang, well, both of them have been really at the forefront of developing better viral tools, which is what we will need for delivery. And in addition, the Blackshaw group was part of a consortium trying to identify networks of genes important for in the retina. And he can, we benefit from things that they learned through these regular conversations. More broadly, there are lots of other efforts to promote regeneration, and we read the literature and we talk about it, and we plan, in fact, to have some more regular direct interactions with people working in regeneration, so that we can apply that effectively. And I know that people like Tatiana have already, where her work in the zebrafish is very granular in terms of what they can see with genes. And already she's been able to, identify kind of these modules of genes that are very familiar based on what happens in other regenerating systems, for example. And so I know that she's been looking a lot at, similar pathways in other regenerating organs. That actually is what makes the consortium so special. So, industry is built on what we can discover in academia, right? And so for industry to be successful, we have to have a very deep understanding of how systems work and just generating the data to be in a position where you can actually say, no, this is the gene that we need to focus on. This is where the drugs are going to come from. Traditionally, people will work on that by getting money through the NIH, but that's critical to the mission, but it leaves out the opportunity for synergy. And so, in addition, a lot of the NIH funding is tied to less risky, more safe bets. And it's important. We need that strong foundation. But I think, we all know just by watching the way science has unfolded over the decades that, we need to make big leaps forward. We need to do some things that maybe, are a little bit risky or are off the wall, out of the box. And that's what our consortium can do, because we can actually discuss these ideas, try something that people haven't tried, and then share it and move everybody forward to the point where industry can benefit. And I think the fact is that this is already happening, because there's a lot of exciting work in gene therapy for deafness, and that work is built on the basic science that has been done in labs. And it's, taken a long time to get there, but now industry can do it. And I think that that's exactly where we will fit in, is we are going to position the world to identify the best way to come up with some effective treatments.
Several of us do work with companies, and the reason to work with them is that they have the infrastructure in place to actually launch treatments and interact with the FDA and go through the very long and very expensive process of bringing treatments to patients. But we see our role here as bringing it right up to that point and going as far as we can. Before doing that, the main way that we communicate is through the gear, which was mentioned before, as a place where we share a lot of the raw data and partially data that's been put into a form where others can use it easily. And then the other way is through publication and at meetings, which are the typical ways that academic science gets communicated. We've had some discussions with industry as a group, and are sort of continually looking for ways that we might collaborate with industry. Their main problem is that these are long-term problems and that they really don't have the long-term commitment from not donors, but investors to take the long view. And so as a result, much of the work done in companies on hearing loss now is being focused on genetic hearing loss, which will be a very important contribution to kids with hearing loss that's genetic, but which doesn't begin to tackle the much larger societal problem of hearing loss, which we call sensory neural hearing loss, which is not directly related to single gene defects.
Typically, evolution works by selection. So the fact that we lost the ability to regenerate hair cells means we must have gained something that is meaningful. And that could be just kind of something to do with the complexity or resilience of the cochlea. So for example, maybe it's maybe the junctions are so strong in the cochlea and that helps us have good, strong hearing, but maybe the presence of those strong junctions makes it hard to rebuild and put in a new cell, something like that. I'd be really curious if any of my colleagues here have additional thoughts about this really interesting question that I wish I could, give a clear, firm answer on, but I think it's hard to know. So if anybody else would like to say something?
I can try. So yes, I agree. We don't know. It's all a matter of speculation. But just like you said, Lisa, we gained a lot. So we gained in acuity, in a huge dynamic range. From in humans is 20 hertz to 20,000, and some animals can hear even higher frequencies. And the mechanism that allows for that to happen cannot be easily replaced once damaged. And I want to add that it's not only humans, it's all mammals that cannot regenerate their hearing hair cells.
And I think Yoash makes an important point here, and it's one of the reasons why we're all happy to be in this room is that he said that humans cannot regenerate their hearing hair cells. In fact, humans can regenerate some hair cells. So in the vestibular system, there is regeneration. And so I think a lot of our goals are oriented around the fact that there is a capacity there that we just need to learn how to tap into and to take what is working in some of the structures in the ear and figure out why it doesn't work in the cochlea. So that gives us a lot of optimism.
Jenny, I want to ask you, you had mentioned in a session this morning about how that you've often heard people say that there's no regeneration in mammals. Mm-hmm. And could you share those conversations and give us some insight on your thinking around that?
Yeah, sure. I work specifically on the vestibular system, as Lisa just alluded to, and that's the other 5 organs in our inner ear that allow us to sense where our head is in space and allow us to move our body with coordination and feel a sense of equilibrium and balance. And remarkably, in the mice that I studied, but also from others' works in humans, it's very clear that there's a natural capacity for regeneration of hundreds of hair cells after damage in these balanced organs. And what's great is that these organs share a kind of an embryonic origin with the cochlea, our hearing organ. They're very similarly structured. They function in essentially the same way. They have connections with the brain in a very similar way. And so given this kind of conserved biology, with these balance organs, it raises a lot of optimism that there is a latent ability for regeneration in the cochlea that we're trying to tap into. And by studying the balance organs, it allows us to get insights really specific to that species. So I think it's important to remember that there is some capacity that we have to repair our hair cells. We just need to tap into it and figure out how to amplify that process, how to kickstart that process in the cochlea.
This is a terrific question, and it does not require a lot of imagination, because we already have an example that happened in the near past, right? Based on studies very similar to work that's done in the consortium, there was hope based on basic science research that simply giving one transcription factor, it's a protein that regulates other genes, introducing it into the ears of adult people with hearing loss, it will result in hearing restoration. And when there was sufficient evidence, according to the Food and Drug Administration, a first-in-human gene delivery trial was approved, and unfortunately, that study was not successful. Now we think we know some of the reasons for why it was not successful, and our hopes are that new combinations that are offered by our consortium will lead to the necessary new evidence that will allow such a study to be successful. The take-home points from that are, however, that gene delivery to human inner ears has been approved by the Food and Drug Administration, that the technology to introduce such genes surgically and safely is available. And in fact, the best examples to look out for in the next 18 months are from basic science, very akin to what is done by the consortium for genetic diseases with a known molecular basis, specifically mutations in the gene otoferlin. There are, based on work that was done in basic science laboratories, just like the ones of the people in this room, resulting in sufficient evidence. There are 3 separate consortia and companies that are competing to be the first in human to restore hearing in the very, very small population of individuals that are born with hearing loss due to mutations in this gene. Nevertheless, if they'll be successful, that will, I think, open the floodgates for many, many more studies to be accepted.
Can I follow up on that? I agree completely with Rona's important points, and there's a lot of reason to be extremely enthusiastic about the tools that are being developed in studying congenital deafness, how they can be applied to promote regeneration. One important difference, though, is that we know what causes congenital deafness. Usually it's a single mutation. So we know what we need to do to genetically manipulate it. As far as regeneration goes, right, we've been working on this for 15 or 20 years. And so it's not a single gene, most likely. It's a set of genes, and it's potentially a set of different manipulations we need to do. And so this is the reason why the consortium is so critical, because we really have to discover what these genes are that need to be manipulated. You know, what their specific functions are. Are there genes that are blocking regeneration? Are there genes that we could silence to allow regeneration to happen? Are there genes that are not turned on in humans that we can turn on? And so one big project that we're doing is this gene discovery, where we're learning about new genes. But in parallel, we're also testing hypotheses about whether these genes are, if we manipulate them in mice, are they enough to promote regeneration? I don't know any other groups around the country that are doing these things in parallel, and there's crosstalk between them, right? If one thing doesn't work, we simply go find out more gene discovery and we maybe apply that. In fact, we were talking about this 2 hours ago. So the other thing I'll say is that, you know, by developing tools as we're doing, such as with gear and other really advanced cutting edge tools to study these things and sharing it with the public around the world, we're helping to accelerate their work as well as ours. And to get something from bench to bedside, it takes this basic work, this basic understanding, the translational approach, so knowing if it's going to work in a mammal, like a mouse or a rat, but then doing the human testing. And so everything we're doing is critical in that pipeline.
Can I add briefly that maybe Rona could not say because she's an ear surgeon and a physician, but I think one of the strengths of the consortium is that we are all scientists, but some of us are also physicians, a person on the board and Rona. And so we're always kept in the focus of how that might be translated at some point.
And maybe I'll also just take the chance to follow up on something very important that Jenny said, which is that we share what we're doing with others. And, the way that scientific communication generally happens is through papers. And so that is one of our big goals, is that right now we've done a lot of analysis, and there are difficult studies to do that could not be done by just one lab, and we are sharing those results. So this paper will be an analysis of genes that are expressed in 28 different kinds of hair cells, like Seth was summarizing before about the working group. So this paper will both will present our method for comparing when you've studied genes in different species using different methods, right? This can be very difficult. And then we've found ways to make these comparisons in a robust and reliable way. So the paper will tell people what our data are, and will give them access to our analysis so they can ask whatever question they want. So for example, if the lab is has identified a new regulator that they think promotes, hair cell development, and they want to see how far they can get and compare it to what we are already doing, they can look at their genes and the genes they find in their analysis and see, well, how do they behave in zebrafish? How do they behave in chicken? And this is something that Rona's working on right now with the gear to make it very simple. So you don't even need to know the anatomy. You can just push a button and say, here is a pathway that I think is important. Let's see how it behaves in the regenerating chicken ear. And so we really want people to realize that this tool is available, and the papers can be the best way to do that.
So if somebody says to you, how do you know the consortium is actually moving forward? How would you answer the question, Rona?
Yeah, I'm happy to take this question because yesterday morning was probably one of my happiest mornings in a long time. And I enjoyed yesterday morning more than our annual meeting a couple of months ago, or essentially any seminar that I've been to the past year. For many years, and I've been on a consortium now for at least 7, we've been trying a lot of things, and we've been talking about what else do we need to do, and what else do we need to look at, and we still do that, but we were always talking about that in face of pictures of really nothing showing up. So we would try these things and everything would look like nothing happened, except in the organisms that normally do have regeneration. And yesterday we had so much level of evidence that regeneration will actually happen in mammalian ears. We had such amazing data presented from the group of Andy Groves on regeneration, albeit not in the right place, but close to it. Well, before we didn't see any of that. Nothing happened. So we're seeing what we want to see. We just need to figure out how to move it to, shift it a little bit to the right place. We saw new tools developed by John Burgandy to be able to more specifically test these things and actually bring the genes to human, because obviously we're not going to genetically engineer the human. So the data are there to show that this consortium is uniquely positioned to be successful.
And I might like add to that. I totally agree. Yesterday was amazing because it was, I think a time where this group really has pivoted from here are the data we want to collect, here are the ideas we want to test, and progress was how close are we getting to having those data to now we're seeing the results. And I think that feels great. And so in terms of, how are those data then represented in kind of progress? It's through publications. It's through once we have those data to say, guess what, this really is possible. You can write a grant to the NIH and bring in more money and expand that work. And in addition, I've mentioned, we're professors, we're training people. We are working with people and giving them the skills they need to continue to work on regeneration. And so every year when I ask for progress, I also ask to find out how the people who are working on these projects are what they're doing. They're joining industry, they're starting labs to work on regeneration. This is another sign of progress, in my opinion.
Plus, we are now thinking about how we hand over the stick to the next generation of researchers. So we need to rejuvenate us. I mean, look at this, no hair, gray hair. We need to bring in the young people. Plus, the technologies that we are now using currently, we haven't thought about these 10 years ago, and we made huge advances just based on the technology. And if you think about what is going on in the world now with artificial intelligence and how we look at data sets, the next 5 years will have another boost of technology development that some of us cannot deal with. And I clearly say that we need to bring in the young people to actually slowly replace us and take over the world. And we can be in an advisory position and can help them to get going. And so doesn't mean that we all drop out in the next 10 years. I think we will slowly transition and bring in fresh energy and really move this forward. Because if you really want to translate this, you need to do this in multiple steps. And we need all these young people that we are training.
And it is very exciting that the number of folks that each of the folks sitting around this table represents a laboratory, and multiple researchers, folks that are either master's degree or PhD candidates, or folks that are working in the lab. So there is an army of folks that are represented by the folks sitting around the table. Jenny, I want to ask you a question. You had mentioned before that someone had asked you, how would you summarize the work that the consortium is doing? And I was wondering if you would share that conversation with us.
Yeah, it's hard. It's hard in a sentence or two to as you notice how complex things are and how wonderful they are, to sort of simplify them. But the support that we scientists get from HHF allows us to work together in ways that have never happened before. Prior to this support, we all worked in silos, and we talked to each other, but we didn't freely share ideas. We didn't freely share criticisms, and we didn't push each other to do new things as much, right? Because we were somewhat in competition, honestly, competition for federal funding. So now we do that. We work together. And I think the strategy that we've approached, or the approach that we've taken, like the strategy that we've adopted, is threefold and extremely unique. And that is that we are brave enough to do this gene discovery across these species that we mentioned, which is a lot of, as Seth said, a lot of big data generation, a lot of really hard work in the lab, but is creating completely new insights into what restricts regeneration and what might allow it. So that there's this gene discovery arm. There's this translational arm, which is really challenging, that the reprogramming group is doing, misexpressing genes in damaged ears to see if they get regeneration. So that's actually the proof of concept of the discovery work is put to test in this translational work. And then in parallel, as we do this work, we're building new tools. We're building new tools for the field to accelerate progress And so I think, the way that this consortium's evolved through the years, through the support of the Scientific Advisory Board and the HHF board and the donors and their input, has just made it a really strong and effective approach to exact change and improve hearing health and maybe balance health for people around the world.
I just want to kind of underscore the message that this is a special opportunity to make a huge difference. And I think that none of us can say exactly, when that will happen. It could, and that's part of the excitement, right? Is that none of us had heard of CRISPR 10 years ago. And that's transformed things. None of us knew that you could keep mRNA in a stable way, and that's transformed human health. And this group is ready to jump the minute there's a chance to do something transformative. And we are actually the ones who are, making that transformation happen, with the group, with the data. And what I feel is that really, if the goal is to find a way to restore hearing, this is the work that has to be done. And if we do not do this work, we will not be able to restore hearing. And so thank you for your support. I think it's immeasurable, like what an impact you've had on the field. Thank you.